move to the transcriptomics part. I'm really excited for this part. So please welcome Professor Jason O'Brien from Wharton University. So if you have everything ready, you can start with your presentation.
All right. Just want to check that you can hear me.
Yep.
Yeah, and I'm going to put my screen in presentation mode here. Are you seeing the full presentation?
We see the presentation mode, like presenter. Yeah.
Presenter
we see
mode?
the
Okay, I'll
notes
just
as
change
well, though.
Yeah, I'll switch that. How's that?
Yeah. Now it's
Better.
working.
All right. A little quicker than usual. All right. So my name is Jason O'Brien. I work for the Ecotoxicology and Wildlife Health Division at Environment and Climate Change Canada. And I'm going to be talking to you about our work in transcriptomic points of departure and efforts to establish best practices for regulatory applications. So, some standard background information that we're all familiar with. There's hundreds of thousands of chemicals registered for commercial and industrial use around the world. There's many inventories in different countries that describe thousands of these chemicals, but these don't even include the unintended byproducts and metabolites and mixtures and so on. So we're exposed to just very, very many chemicals, and very few of these have been thoroughly evaluated for human and environmental safety. And one major reason for this is there's a major bottleneck in toxicity testing. The traditional methods of toxicity testing just can't keep pace with the amount of chemicals that we need to regulate. The methods are slow, expensive, they use a lot of animals, and they're generally not that effective. They're inconsistently predictive across species, and this is because they're largely based on high doses and acute lethality. And so as a result, only a small fraction of these chemicals have been comprehensively assessed for safety. At Environment Canada, the basic paradigm for quantitative risk assessment is to derive a risk quotient that compares exposure levels to concentrations that cause hazardous effects. When this ratio is less than 1, generally that means the exposure is less than the dose that causes hazard, so it's assumed that there's no risk for the chemical that's being investigated. But when this ratio is greater than 1, it indicates a potential risk. Now, this is a large oversimplification, but it gives the general idea of how it's done. For exposure levels, this is generally estimated from monitoring, but more often from modeling data. And this is done to estimate predicted environmental concentration. And the hazard component is derived from animal exposure experiments, where there's a dose response type study where we get a point of departure that's used to estimate a predicted no effect concentration. And as we just talked about, the traditional animal testing methods are very expensive and time consuming and not necessarily effective. And so there's been a lot of effort to develop so-called new approach methodologies, or NAMs. These are technologies that can increase the efficiency of toxicity testing and reduce our reliance on animals use. There's many prominent categories of examples of NAMs. There's the high throughput technologies like in vitro methods, animal alternatives, miniaturization and automation. There's the computational toxicology, QSARs, modeling, machine learning, and AI. And of course, there's the high content technologies that we'll be talking about today. And in particular, I'm going to be looking at how genomics can be used. Now, one of the reasons that genomics technologies and omics technologies in general are so attractive to use for toxicity testing is because molecular changes can be detected at much earlier time points compared to traditional apical effects. Changes in gene expression in the transcriptome, for example, can be detected within hours or days after an exposure, in contrast to traditional endpoints like mortality or cancer, which can take months to years to manifest. And so the methods can be much more quick compared to traditional methods. Now, the promise of toxicogenomics, of course, has been around for quite some time, but to date, genomics has not really been used quantitatively in chemical risk assessment. It's been used more in a qualitative way, if at all. And this is largely because the kind of classic ways of doing genomics research doesn't really fit into this traditional risk assessment paradigm that I showed you before. Doing clustering analysis and network analysis doesn't really fit into this quantitative risk quotient calculation. And so it's been a little elusive trying to figure out exactly how genomics can be used in quantitative risk assessment. But what I'm going to be talking about today is how a transcriptomic point of departure can be used to estimate a predicted no-effect concentration that can be used in quantitative risk assessment. So what exactly is a transcriptomic point of departure? The traditional endpoints are usually evaluated with a dose response analysis. We saw a lot of examples in the last talk, and I'm sure we'll see lots more in the talks to come. And the general goal is to determine at what dose a compound starts to have an effect on an organism. And this is known as a point of departure. Some of the more common points of departure in toxicology and risk assessment are, for example, the 50% effective concentration, EC50. There's also the Lowell and Knoll, which are often used. And more often these days, we saw some examples just moments ago of the benchmark dose, which selects a predetermined benchmark response, often 10% or 1 standard deviation above controls is a common benchmark response, and that's used to derive a benchmark dose. But one of the challenges with using this approach in genomics is that dose response analysis requires a large number of dose groups and replicates, which has not been very practical in genomics until recently. But recent advances in DNA sequencing have really dropped the price. And really, that's all it really is in conducting genomics experiments. And now, because the prices have gotten so low, we're down to doing whole transcriptomes for less than $50 a sample. And it just keeps going lower and lower and lower. And now, so this allows advanced experimental designs, larger sample sizes, larger number of doses, even different time points. And so this makes genomic dose response analysis economically feasible in a regulatory testing capacity. And so just like the traditional endpoints, transcriptomic point of departure, or TPOD, is an estimate of the lowest dose for a significant change in gene expression. And what that looks like is if we look at the expression of a single gene and look at it, how it responds across a range of doses, we can fit a curve to it and select a benchmark response, perhaps 1 standard deviation above controls, and derive a benchmark dose, just like any other endpoint. What's different is that this is repeated for every gene in the transcriptome, and what we end up with is hundreds, thousands of dose response curves and associated benchmark doses. And so how do we derive a single genome-wide transcriptomic point of departure from this? Well, there's a couple of ways to do this. The two general methods, the first one is the distribution-based or pathway agnostic method. We just look at the distribution of all of these BMDs across the dose range. And if you can think of a BMD as kind of the dose at which a gene gets turned on or turned off by a compound. And in the example here, if we look at the dose between the range of 1 and 10, there's a large number of genes that get turned on, and we would consider this a transcriptomic point of departure using the distribution-based method. Another method is to use the pathway-based method, where genes are grouped into pathways, and then we look at the range of different BMDs within the pathway, and we sort those pathways from least to most sensitive. And very often, the median BMD of the most sensitive pathway is set as the transcriptomic point of departure. So these are two different ways to do it. They generally get you a very similar response, and each of these kind of approaches have multiple methods within themselves. So there's lots of different ways to do it, but generally they get you very, very similar results. And what they all kind of represent is a dose below which there's no concerted molecular change detected, and therefore adverse effects are unlikely at these doses. And as I mentioned, molecular changes can be detected much, much earlier than traditional time points. So the question of the relationship between these early points of departure versus traditional points of departure became of interest, and we wanted to know, can TPODs from short-term studies be used to estimate doses that cause long-term toxic effects? And some pioneering work in the field done by Russell Thomas from this 2013 study showed that a transcriptomic point of departure from a 13-week exposure was highly correlated to a traditional point of departure from a 2-year cancer bioassay. It took 2 years. So after 13 weeks, you get the same quantitative value as you would from a 2-year study. And this was shown in both rat and mouse. So this was pretty exciting in the field. And since then, a lot of people have repeated similar experiments and showed similar outcomes. At Environment Canada, we conducted a literature review of over 100 such articles, and we found that this correlation between a short-term transcriptomic point of departure and long-term traditional point of departure was very strong across all studies. Generally, there's a slope of nearly 1 with an R-squared of 0.8 for all studies. 95% of the TPODs were within 10-fold of the traditional point of departure. Most of them are within 5-fold. And this correlation existed even after very, very short exposure periods. Even at 1 to 5 days, there was a strong correlation between a transcriptomic point of departure and a long-term toxicity test. Now, most of this evidence comes from rodent data. We were also interested to see if this held true in ecological species. When we started this investigation only 6 years ago, there weren't very many ecotox relevant data sets. We only found 12 where we could do this sort of analysis. That number has increased since then, but it's still generally mostly driven by rodent evidence. But we did an analysis of these 12 data sets that we found, looked at a variety of species, fish species, frog, Daphnia, different life stages, different exposures, variety of tissues, genomics platforms, and to a variety of different chemicals. And when we did the analysis, we got our BMD histograms to derive our point of departure for all of these compounds. And when we compared them to traditional chronic LOX, we found, again, a very strong relationship between a short-term test using transcriptomics and a more traditional chronic LOX. So very encouraging evidence here. And I think since this was done, more and more evidence emerges that shows similar results in the ecotox realm. And so because this evidence is suggesting that this might be have regulatory applications, quantitative risk assessment, there's been a lot of efforts to establish best practices to enable to use this in a regulatory setting. One of the first groups to do this was the U.S. National Toxicology Program, where they published their approach for genomic dose response modeling in 2018. This was kind of the first standardized method for deriving TPODs. It was based on an expert panel review of the methodology in the literature. And from this exercise, they made recommendations for experimental design, standard data analysis pipeline, how to interpret the results. And they developed the software called BMD Express, which is still kind of considered the gold standard software for doing TPOD analysis. And this has been kind of considered a guidance document that many people use when they're designing their TPOD studies. At the international level, there's also been a lot of efforts to kind of harmonize approaches to using this. One of the groups that I'm involved with is the Health and Environmental Science Institute E-STAR Committee. HESI is a nonprofit organization that brings together scientists from academia, government, and industry to address global health and environmental challenges. And the E-STAR Committee specifically looks to develop innovative approaches for toxicology and risk assessment. And they have a variety of working groups, including one for looking specifically at using the omics point of departure. In that group, we recently published a paper describing a vision for using transcriptomics in regulatory chemical testing. And in that paper, I won't go through all of the details here, but we describe a logic framework for how the evidence needs to be organized in order to support the use of transcriptomics for risk assessment. And we concluded that largely the evidence exists to support all of these principles, and transcriptomics technology is basically ready to use for regulatory applications. Of course, we'll go into more details if you're interested in this paper. Some of the major challenges that needed to be overcome for using genomics in risk assessment are standardization, transparent reporting to promote reproducibility. And standardization in genomics and bioinformatics research is a lot like herding cats. It was a major challenge, but it's something that really needed to be done and demonstrated. And some of the first efforts in this area, important efforts, were the development of the RODAF, the Omics Data Analysis Framework for Regulatory Applications. Most people kind of have accepted that if you give two different bioinformaticians the same data set, they don't necessarily always come to the same conclusions. And so the RODAF is meant to be a kind of standardized method of processing your data all the way from your raw transcriptomic reads up to your differentially expressed genes. Includes some filtering steps and some quality control steps. And I shouldn't say it's a standardized approach. People, the genomics and bioinformaticians don't like to hear that word. It's considered a reference baseline. So you can conduct this analysis, and it's a baseline to compare to any novel method that you might have. So any new method that you develop should at least be as good as this method, and it can be used to compare any new methods. And another important thing about this method is that it's not a black box. It's completely open, freely available, and based on freely available software. So it's very transparent on how it's done, and anybody can do it. Taking a closer look, you can see that over on the right side here, we have some describes all the different steps. It tells exactly which tools to use for all the different components, different parameters, and some QC thresholds that need to be met. And we've been using this with a lot of success in our group, but we have encountered a few challenges. One of the challenges is that the RODAF, as described right now, is very technology specific. It's described currently only for Illumina-based RNA sequencing and microarrays. And so you kind of have to redevelop it a little bit and modify it for any new technologies that come along. For example, TempoSeq or 3' sequencing, which is what we use in our lab. Another challenge is that this approach only goes up to deriving your list of differentially expressed genes. It doesn't describe any downstream analysis like doing dose response analysis or how to interpret those results. And so this is where the HESI eSTAR Committee TPOD group has focused their efforts on currently. We just published this paper describing the bioinformatic workflows for deriving TPODs, where we reviewed the current state of the bioinformatics, we identified variations in the pipeline and make recommendations and identify problematic areas. And we actually have a ongoing workshop right now that's running. It started in January, and we're planning on running it until October, where we hope to address all of these data gaps and make recommendations on some baseline or standardized baseline approaches, and maybe recommend some experiments that can be done to address any outstanding issues. So this is ongoing right now. In addition to describing standardized pipelines for analysis, reproducible pipelines, an important component of genomics research is reporting. A lot of the times in the literature, it's hard to recreate an analysis based on just what's reported in a paper. And so this issue was taken on by the OECD, and they developed an omics reporting framework, the ORF. This is a reporting framework that tells you how to describe in detail all the methods that you're using to help ensure that all the information required to understand, interpret, evaluate, and reproduce an omics experiment are available. And the way that the reporting framework works is that it's a modular framework. There's different components to describe different parts of a genomics experiment. There's components to describe the experimental aspects, and then there's some technology-specific modules for microarrays, RNA-seq, even for mass spec and NMR spec. And there's some data analysis modules. So when you do a genomics experiment, you fill in the reporting framework so that people can reproduce your results. And this the ORF was put through some trials to see how well it worked, where there was a submitter that produced a data set and did some data analysis, and they submitted their results to a referee, and that referee passed those results, the raw data, to an end user along with the completed reporting framework, and the end user tried to reproduce the original submitter's results. And so here's one one of those trials results where you could see that the end user and the submitter had the exact same number of differentially expressed genes at the end of their analysis, and that the fold change values were completely exactly correlated. And this type of reproducibility in genomics research prior to this was almost unheard of. So this is a big success for the use of this. And it was even used in a trial for dose response modeling, where the submitter generated a bunch of TPOD data. I won't go through all of these different values here, but they're all different types of TPODs. And the end user tried to reproduce these results under a variety of different scenarios. And in general, they got almost exactly the same results. There's a few instances where the end user got a few slightly different values, but these are actually expected because there's some random processes involved. And so it's not unusual for things to be off by a fraction. So again, a huge success demonstrating that these results can indeed be reproduced if they're reported correctly. And so all of these efforts that have been ongoing have started to build the confidence of using this technology for regulatory applications, and actual regulatory uses are beginning to emerge. And one of the biggest milestones in this area of research was last year when the US EPA released their transcriptomics assessment product, or ETAP. This is a transcriptomic-based product that is intended to use for data-poor substances when there's no other existing repeat dose toxicity studies. It's based on a 5-day in vivo repeat exposure in rodents. They use 8 dose levels with 4 males and 4 females per group, and they look at gene expression profiles in 12 different tissues, and it's based on the BioSpider TempoSeq genomics platform. And what they end up with is what they call a transcriptomics-based reference value, which is based on the TPOD methodology that I showed you earlier. And what it is, it's an estimate of the daily oral dose to the human population that is likely to be without appreciable risk. And the very first one of these used at an actual risk assessment was used for a PFAS compound called MOPA just last year. So this is a massive success in our field, and we were so happy when this was done. Now in Canada, and you'll hear a lot more about this from Dan Villeneuve's talk tomorrow. I think he's the first one to go. In Canada, we're not quite there yet, but there's a lot of very exciting work in the area. Right now, we're working with McGill University and Genome Quebec under this Genomics Applications Partnership Program. And the goal of this program is to validate the use of TPODs using fish embryo models for ecological risk assessment. This approach is based on the 3' RNA sequencing, and we also use EcoTox chips, which are 384-well qPCR arrays, which were developed at McGill University. We're using 3 different animal alternative fish embryo models: fathead minnow, rainbow trout, and zebrafish. And we've also developed a user-friendly data analysis platform. We're currently in the first year of the 3-year funding. And so I hope next time I come here, I'll be able to share a lot more with you about this. But we're definitely looking at bringing this into risk assessment, quantitative risk assessment in the eco realm at Environment Canada. Now, there's some other challenges that are still outstanding. I don't have a whole lot of time to talk about them, but I thought I'd bring them to your attention a little bit. There's a strong desire to bring this technology to in vitro analysis, but this will involve, some in vitro to in vivo extrapolation and some other complications that come along with that. And though I don't have time to talk about it in detail, I would highly recommend you check out this paper from Anthony Reardon from Health Canada, where they discuss vision towards best practices of using in vitro for risk assessment. And invariably, whenever we talk about the transcriptomics and points of departures, it always comes up about understanding the underlying mechanisms. And currently, the methods used for transcriptomic points of departure are agnostic to the nature of the biological process underlying the TPODs. Simply put, these approaches aim to identify the lowest dose at which a biological alteration are expected to occur, but not what those adverse effects are likely to be in the long term. And this has always been a sticking point for some risk assessors who feel that there's a need to clearly identify what a hazard is before you can regulate that. But the approach that we're taking is that it's much more valuable and possible right now to determine a dose that is protective very rapidly and efficiently, and you can achieve your protection goals without necessarily predicting the exact mechanisms and the exact outcomes that could occur. And so, but that being said, there's a lot of progress being made in this realm where our ability to make predictions about the mechanisms are definitely improving. And I'm sure we're going to be talking a lot about that in the sessions to come in this workshop. And so in summary, transcriptomic points of departure from short-term studies have been shown to be protective of long-term toxicity. There's a large body of evidence supporting this in rodent models. There's some evidence in eco-relevant models, but there's definitely a need to start making these relationships much more strongly supported in the eco realm. There's been a lot of efforts to establish best practices. The NTP, of course, has their published method. And there's been a lot of work in the bioinformatics area where some reference data analysis pipelines have been developed and some reporting frameworks have been created. And this has all helped to increase the confidence about the reproducibility of this technology. And because of this increase in confidence and the strong evidence supporting this, there's been some regulatory applications that are starting to emerge, the first of which is the US EPA's ETAP, and hopefully Environment Canada and other countries are not too far behind. That, I'll take any questions.
Thank you, Jay.